The brain is an input-output machine. So senses, so like the things that we see and hear, they bring in information, and then our brain is able to process that. And then the actions that we do are the output. So basically, I might hear something, my brain processes it, and then maybe I say something back. That's the output. And so the brain takes stuff in, and then stuff comes out. The brain is comprised of 100 billion neurons and other cells. And because there's so many cells and it's so complicated, it also means that it can do a lot of really varied functions. And so that's why we can do lots of things with our brain. And the brain is dynamic, and it changes itself, and that's how we can learn and remember things. And so the brain will change itself on the order of seconds, minutes, hours, days, years, and so it's changing all the time. So I'll be talking a little bit about what are neurons. So I said neurons earlier, but what neurons are the cells in the brain. So the specialized cells in the nervous system, and they look kind of like this. This is a cartoonish diagram. And they have three main structural components. So they have these things called dendrites, which are the little things that kind of protrude out, and that's where the information comes into a neuron. There's the cell body, which is where the information is integrated, meaning like that's where all the information comes together and is processed. And then there's the axon, which is the output. So basically information comes in through the dendrites, the cell body processes it like a computer, and then it's sent, and the output is sent down the axon. And so an incoming signal comes in that side of the neuron, and then the outgoing signal goes out on that side of the neuron. So the information flows that direction. I don't know if I have a laser pointer, but hopefully you guys can see it come up on the screen. Neurons are excitable, and excitable basically just means that they're electrically active, and they signal using the flow of positive and negative charges. So kind of like a battery, how a battery has a positive and negative end. There's positive and negative charges swimming all around in the neuron that help it communicate with other neurons. And the reason that we use electrical signaling is because electrical signaling is really, really fast. It's the fastest signaling in the body. And neurons use something called action potentials as a signal. And what an action potential is, it's a really, really quick electrical spike. And diversity in neurons is expressed as diversity in their action potentials. So different neurons have different action potentials that look very different. And so it's like kind of like different, sizes of gears or different ways that things turn to make things work. So you can kind of see, this is a very scientific graph, but basically all I want you to see is that the spikes are different widths. And so that means that the neurons do different things. Neurons have ion channels in their membrane to help make electrical signals. And sodium channels are a type of ion channel. So they're ion channels that let sodium pass through. And here's a diagram of the neuron, and the sodium channels are present all throughout the neuron, but you can kind of see them in that little zoomed in section, and they let sodium into the cell all the way down that neuron. And sodium channels are very important for the neuron signaling called action potentials. Neurons function in networks, so they're all connected together and they constantly talk to each other, and those networks are really important when it comes to things like seizures and like epilepsy. And so seizures can typically result from brain activity in that network that's both hyperexcitable and hypersynchronous, meaning that there's a little bit too much activity or it happens all at the same time. The brain has to achieve and maintain a really precise balance of excitability. I kind of think of it like a teeter-totter, and you don't want the E to get all the way to the top. But it's a constant balance for the neuron of excitation and inhibition. And so doing that, messing with that balance a little bit, unfortunately leads to seizures. Or if you mess with balance the other way, it might lead to something like intellectual disability. And so unfortunately, that's what's happening with SCN8A variants. And so when we're talking about research, we're of course thinking about these seizures that can result from brain activity that's hyperexcitable and hypersynchronous, and we're thinking about the really precise balance of excitability and inhibition. And so that kind of leads to the major aims of epilepsy research, which are how do neurons become hyperexcitable? How do they become hypersynchronous? And how do we get that balance back? And how do we get the neurons to start to behave normally again? Yeah, I think there's a question.
How would this relate to like auditory and visual processing in the SCN8A of like using colors to pinpoint? Is this where that processing issue lies with possibly?
Sort of, yeah. So the question, if everyone didn't hear, was how does this relate to things like auditory and visual processing with SCN8A, and maybe issues with processing, and maybe sometimes also things like visual or auditory cues can like trigger a seizure. So this is sort of related to that. A lot of times what might happen is that a visual or auditory something in the environment would maybe like trigger a cell to fire, and that kind of sends it down kind of a negative cascade that, you know, this cell fired and then it tells all these cells to fire. And so, yeah, it can be related to that. So it might basically trigger the network that's already there. And then in terms of the processing deficits, so like cortical visual impairment, yeah. So this is a little bit different from that. That is more directly related to the sodium channel function in the retina and not as much the excitability. I'm a little bit talking more about how the cells function together, but that's more of a just sodium channel specific deficit, and also a developmental deficit sometimes because SCN8A helps control how the brain and the retina and the whole nervous system develops. It can develop differently when there's a mutation. Awesome. So we're going to get into a bit of kind of hard science, but I want to make sure that we understand kind of what's going on. Yeah. I don't know if I know how to do that, but yes, I can. Mm-hmm. Mm-hmm.
We kind of know where is this all going, why is this all happening. So what specifically is your goal?
Yeah, so I'm not doing research right now, but what I was doing, so there are a whole bunch of different types of cells in the brain, and it's not just cells that are excitatory. So when I go back to that kind of teeter-totter, there are a lot of cells that do excitation, and so they tell things to go. There's also cells in the brain that almost work like car brakes and tell things to stop. And then there's a whole bunch of other cells that also express SCN8A that help with that whole process. So I worked with both the brake cells and then the helper cells. And so what I did with my research was seeing basically there's a bit of an issue with how the brakes basically are failing. And so it's not only just that, you know, the theoretical gas pedal is pressed, it's also that the brakes are failing. So because SCN8A is so, it's a really, really important sodium channel, it is kind of everywhere in the brain, and it impacts a lot of different types of cells. And I realized I didn't repeat that question, but he was asking a little bit about my personal research and how, you know, we kind of know that the cells become hyperexcitable, but where do we go beyond that is kind of what he was asking, I think. All right. I'll talk a little bit about action potentials. And so that little spike there is what we call an action potential. It consists of 3 different parts. The resting phase, which is that green phase, you can see that the neuron, which is what it's tracking, is just kind of resting there. It's flat. And the memory potential is which is what we call that voltage. It's stable. The rising phase, and you can see in that line, the cell increases in voltage really, really quickly. And so that's the memory potential. It rises really, really fast. And then the falling phase, you can see that everything falls really quickly too. So there's 3 different phases, and this is sort of just a breakdown of how neurons talk to each other. So like, if they're not talking to each other, they're in that green phase, nothing is happening. But then a neuron starts to get some signals from another neuron, and it starts to rise really fast, and then the neuron kind of hits a peak and it goes back down. And I'll explain how that works in terms of sodium channels, but I just wanted to kind of break that down before we got into the sodium channels of it all. So there's something called a sodium-potassium pump, and it's sort of like a battery charger. So I mentioned that these cells run on electrical signals, and so what this sodium-potassium pump does is it charges up that battery and gets the cell ready to do that electrical signaling and fire that action potential. And so it kind of looks like this. So sodium's gonna get pumped outside the cell, and potassium's gonna get pumped inside the cell. And you can watch the little animation. So it's basically just pumping one in and one out. And now you can see there's a ton of sodium on the outside, and there's a ton of potassium on the inside. So now the cell is ready to be excited. And this sets up a gradient. And if you guys remember from like grade school biology, a lot of times things like to follow gradients. So like if there's a lot of water on one side of something, the water goes to the other side. It's kind of the same thing with sodium. So it follows that gradient, and it really wants to go inside the cell. So voltage-gated sodium channels have 3 different states. They can be closed, open, or inactivated. And I'm going to break down those 3 different states. They have 2 different gates. If you can see on the open channel, there is a gate that is sort of more like a ball and chain, and then one that is sort of just an open-closed. So that open-closed gate is the activation gate, and the ball and chain gate is the inactivation gate. So those 2 gates help the channel switch from the open, closed, and inactivated states. And so when the channel is closed, no sodium can get in. And so as much as it wants to, it's just not able to. When it's open, sodium can pass through pretty readily. And then when it's inactivated, it can't pass through again. And so that's sort of what sets up for that action potential that I've been showing you, is that at only certain times the channel can open and close, and that's what lets that membrane potential really increase or decrease. So going back to the action potential, the channel starts out closed, and that's why the cell is resting. That's why it's really, really flat. When it swaps to open, we're in the rising phase. So all the sodium rushes in, and the cell's membrane potential goes up really, really fast. And that's called activation. So the sodium channel activates, it goes from closed to open, and it's ready to basically receive signals, and it's ready to talk to another neuron. And then once it gets a little bit too excited, it changes from open to inactivated. So it's called inactivation. And so that's when it's in that little red-orange stage, and the sodium can't get through again. Does anyone have any questions about that? Yes.
Hold on.
Oh.
In the inactivated state.
Use the mic.
Yeah, hi. For the inactivated state, is there a difference between the fast inactivation and slow inactivation?
Yes.
Are those the same state?
No, they are different. We should probably talk about that afterwards. It's not in the presentation because it's a lot of complicated like sodium channel kinetics and things like that. But yes, there is, and some variants might affect the fast inactivation and some might affect the slow. So yeah. And then there's a question right behind you, Shelley.
So my question was, like, how does it relate to the different phenotypes? Like, is there a
Yes.
inverse reaction there?
Yeah, and so I have a slide on that in a little bit, but I'll give you a bit of an answer now. Different variants in different locations of the channel might affect how it interacts basically with those gates. So some might not be able to totally inactivate properly. Some might open late, or it affects basically how that channel works, and that's why there's either more or less sodium passing through the channel, and that's gain or loss of function. But that's coming in the slides too. And so that's coming in the slides right now, actually. The next bit is how does this all this basic neuroscience, how does it relate to SCN8A? And so a very basic overview of what is SCN8A. So SCN8A encodes one of these voltage-gated sodium channels, and we call it NAV 1.6. So if you hear me or a researcher later tonight talking about NAV 1.6, they're talking about the sodium channel that SCN8A codes for. So SCN8A basically just contains some instructions that build the sodium channel called Nav1.6, and it builds it in the brain. And so our DNA creates components that build sodium channels. And then what causes SCN8A variants? So we all have DNA in our bodies, and it's made up of bases. So one of those is called a base pair. We have C, T, A, and G, and they're the building blocks for DNA. Sometimes when the DNA is replicating itself, when there's an embryo or sometime early in development, it can sometimes make an error, and it's usually just one base in the DNA called a single base mutation, and it results in a different base pair that results in an SCN8A variant. And one base change can lead to a change in the whole protein. And so if you look at this example, if we have a base without the change, it would lead to something called an amino acid, which is basically like the puzzle pieces of a protein, and it leads to a protein. But the base with a change might lead to a different amino acid, which leads to a variant protein, and that affects how the protein functions, how it folds, and what it does in the body. And so this can lead to SCN8A-related disorders. And different variants are different severities based off of how it's changed from the original base pair. And so there's lots of different types of variants. And that kind of segues into SCN8A-related disorders. So variants can be gain of function, loss of function, or mixed function. And typically, but not always, gain of function variants lead to developmental and epileptic encephalopathy, and loss of function variants lead to intellectual disabilities and sometimes absence seizures. But again, that's not for everyone. Sometimes loss of function variants do lead to seizures and epileptic encephalopathy, and sometimes gain of function variants don't lead to developmental and epileptic encephalopathy. Sometimes they're more mild and just lead to like learning disabilities and things like that. But here is a diagram of a lot of the SCN8A mutations. It's not all of them, but it's a lot of them. And so you can kind of see there's a bunch of gain of function and there's a bunch of loss of function. We made this for a textbook chapter we're working on with SCN8A. So if you want it, I can send it to you. And it's just a diagram of what the channel looks like. And so if you see your child's variant, if it's in the yellow region, that's called the voltage sensing region. And it could be in one of the intracellular linkers, which are like the little lines, but we can always go over this later if you want to. So in terms of typical sodium channel function, I talked about closed, open, and inactivated, and how that interacts with the action potential. So just going over it again, when it's closed, everything's kind of flat. When it's open, sodium will rush in. And then when it's inactivated, sodium can't rush in anymore, so it kind of flattens out. When we're thinking about gain of function variants, there's more sodium that passes through the channel, and so that could be through a couple different ways. But overall, it results usually in increased neuron activity. And then when we're thinking of loss of function variants, less sodium can pass through that channel, and that usually results in decreased neuron activity. And so here's just kind of like an example to think about. So we have our closed, open, and inactivated state, and we have our action potential. If, for example, we had an early opening, more sodium would be passing through the channel, and that would be a gain of function. If we had a late opening, less sodium would be passing through the channel, and that's a loss of function. And then I'll talk a little bit about Nav1.6 and why it's so important in the brain. So Nav1.6, or SCN8A, is present at the regions where action potentials, the brain signaling, is generated and where it's propagated, meaning where it starts and where it goes all the way down the neuron. So you can see from that diagram that it's present all the way down the neuron, and it helps the neuron send that signal all the way down to the end. So it helps determine when to fire an action potential. It helps determine how many action potentials, and it determines the shape of the action potential. So it's really, really important. And those are the major components of excitability for each neuron.
Can you Can you please explain the slide once again?
Yeah, for sure. So yeah, I'm just talking about basically the uniqueness of NaV1.6 and SCN8A and why it is so important in the brain, and unfortunately why the mutations do lead to SCN8A related disorders. So it's really important because it helps tell the neurons how to communicate with each other, when to communicate with each other, and it basically how much they communicate with each other and what it looks like. So I mentioned earlier that neuron diversity is expressed by the shape of their action potentials. So this can change the shape of their action potentials. It can change how many times they send a signal down their axon, and it can change when it happens. So basically, NaV1.6 is really important in all of those brain communication signals, and so changing it has very varied effects based on how the variant expresses. Yes?
Is this imbalance between the sodium and potassium able to be seen in like a CBC? Because especially for those loss of function who have related like cardiac issues and stuff like that, I mean, those levels are always wrong per se, but is there like a correlation between the SCN8A sodium channel blocker issue and it showing up in their labs because of that imbalance?
Yeah, so SCN8A is really, it's kind of spread out all around the body. It's in the brain, but it's also down the spinal cord, and it's also present in like even the gut, a little bit in the heart and things like that. So because it is kind of everywhere, it does relate to things like cardiac function and sodium channels, not necessarily just SCN8A, but sodium channels do control like heart rhythms. So yeah, it does relate to all those things that you just mentioned. All right, I want to make sure we're all good. Okay, great. Yeah, please feel free to ask questions. I know I do talk fast. I'm really trying to talk a little slower. But if you have questions, I'm really happy to answer them. Here I'm showing an example gain-of-function mutation. And so a lot of gain-of-function mutations have improper inactivation, which means that they don't fully close when they're supposed to. And so the sodium can kind of leak out. And that leads to basically, instead of going back to being flat, they end up a little bit different than the typical functioning sodium channel. And that leads to sort of differences in the way that the neurons communicate with each other, because basically now their baseline is different. So the way that they start out is a little bit different, and so it affects how they talk to each other afterwards. And that's because sodium can leak through the channel when it's not supposed to. And so going back to kind of the uniqueness of NaV1.6, NaV1.6 or SCN8A determines when to fire an action potential. So in this example with a gain-of-function variant, it will fire earlier than a typical counterpart. And so that might lead to something like a seizure or another adverse event because it signals earlier, and it might set other cells up to signal earlier. It determines how many action potentials, so it often can, if a cell is firing, it makes it fire more. And then the shape of an action potential, so action potentials are typically just one spike, but sometimes because of the excess sodium, it almost looks like two spikes because the sodium keeps leaking through. And so it's almost the reverse for loss of function, where you might have less action potentials or a later fire, but it's similar in terms of how it works.
Raquel?
Yeah.
What does a spike actually mean?
A spike is just a brain sending a signal down. So it's basically like if you think about when you try to talk to someone, it's basically like the brain is saying some words, but it's almost like it's playing telephone down the neuron. And so it's sending that signal down. And maybe if you try to do it a little bit too fast, if you try to talk too fast in telephone, it doesn't send quite right. And so their neurons are just trying to talk to each other. And so in summary, and super happy to take more questions before we go into talking about gene therapies. But the brain is really complex, and it uses these electrical signals to quickly take in sensations, process the info, and execute actions. The neurons are excitable cells that communicate through action potentials, which is basically like neurons talking to each other, and the brain talking to each other inside of your head. And voltage-gated sodium channels are specialized, and they're supposed to initiate and propagate action potentials, so they're starting the signal and sending it down the line. And SCN8A is the gene for NAV1.6, which is a really critical sodium channel for lots of aspects of neuronal signaling. And I'm just thanking Eric Wengert, who's done this previously, because he sent me his slides and I adapted them, and then Manoj Patel, who's also done this, and someone who helped me, my intern, who helped me develop some graphics. But if you have questions, we have lots of time for them. So thank you.
So, I don't like that. Sorry. I know that you'll probably go over like movement disorders at some point, but we're kind of feeling at a loss with my daughter's ataxia. So she's loss of function, but she does have some features of gain of function, more on the mild side of things. But with the ataxia, it's constant, but she's going through episodes where the ataxia is significantly worse, and we're trying to figure out, right, if she's having a different type of seizure, or if she's having like a migraine episode, because she'll have the slurred speech. The ataxia will be significantly worse to where she will fall over, drooping of the face sometimes. Like, there's just so, we don't know, is the ataxia going to get worse because of the SCN8A? I don't know. Help?
Yeah, I can't super answer it because I don't know if the ataxia is going to get worse. Sometimes, all kinds of things, and I know a lot of folks struggle with things like sleep and, that can affect movement disorders and things like that. And, the medications that she's on, if they're being changed and what medications they are, because especially if she is more of a mixed function, it's hard to find medications that work for mixed function because so many of them are sodium channel blockers. And it's sort of difficult. It's hard to answer in this context. And I'm not a doctor. But I'm happy to talk to you after, and we can talk about the variant and things like that. Back there.
What about the mixed types, gain of function and loss of function?
Sorry, I only caught part of that.
What about types that have a little bit of both, gain of function and loss of function? We've never been told exactly what my daughter's problem is. Assumably, it's gain of function, but she has a little bit of both.
Yeah, so unfortunately right now, we only really know if something is gain or loss if someone goes in the lab and takes the variant and they characterize it. And so it's hard to know. We can kind of predict based off of what happens clinically. But one of the things that we're trying to do is characterize more of the variants, because that'll help us with trying to figure out better medications. When it's a mixed function, sometimes, how there's those two gates, sometimes they're impacted a little bit differently. And so maybe one opens earlier, but the other one closes earlier, and they both do opposite effects. So if one opens earlier and the other closes earlier, that's a mixed function. So it's kind of counterintuitive, but different mutations or different variants all have different effects in terms of that. And we actually have one of the things I've been working on in the small research that I am still doing is putting all of that information in one place, because it's not been there so far. And we're learning a lot more about which variants are mixed function rather than gain or loss. We're training a model on that. I think one of the presenters is going to talk a little bit about that tonight. Oh, we have another room here.
Where am I going?
So as far as like the different loops,
Yeah.
Are you starting to find a correlation between the loops of which function, like loss of function, gain of function? That center section, is it mostly gain of function? There are a few dots on the map, but is there more of a distinct correlation now?
So it's not, it is partially, we have regions of the channel that we know are mostly benign. We have regions that we know are likely embryonic lethal. And we also have regions that result in SCN8A related disorders. But within the regions that result in SCN8A related disorders, it can be really varied based off of the amino acid, like the protein building block, like their substitution. So if it's a really small amino acid to a really big one, it's usually more severe. And those amino acids usually have charges. So if it's a non-charged to a non-charged, it's less severe than it is a charge to a different charge, or a charge to a non-charged. So basically, there are regions that usually cause SCN8A related disorders, but within the regions, there's a lot of variability.
So if we know where our star is on the map in theory, like,
Yes.
could you say this is, because if I'm looking at the numbers and whatnot, I don't know if that's a big difference or a small difference.
Yeah.
You know, like, how does that correlate to us looking at our variant and where it falls on the map, like, to actually understand our specific variant kiddo?
Yeah, we should, we can talk later. Yes, and I can talk about your variant. Also, one thing I was going to mention, I was going to get through the slide, but Dr. Michael Hammer, who's usually here, he's not here this year, unfortunately, but he's offering one-on-one meetings, which I'll mention in a second. And he's a geneticist, and he can tell way more about genetics than I can, but I'm, what I am is a sodium channel biologist. So I understand how the channel works, but he's a lot better with the genetics, so we kind of work well together. Okay, I'll mention a couple more things, and then we'll get more questions maybe, and we'll go into some gene therapy discussions. So something I really wanted to mention, because it's really important, is this thing called the KeySet trial. It is a clinical trial that's being done all around the US for patients who don't respond to traditional seizure rescue medications like benzodiazepines, like Valtoco and Klonopin and things like that. And it involves treatment with Keppra and ketamine. And so the reason why I wanted to bring it up is because I know a lot of SCN8A patients don't do well on Keppra. And so this is a trial that you need to opt out of rather than opt in. And there'll be representatives from the trial here during the cocktail hour to help out with that process. But we just, we want to make sure that SCN8A kids don't get Keppra unless they tolerate it well. And so those folks will be here a little bit later to help the opt-out process.
What do you mean it's opt-out? Like, are we all in by default?
Yeah, so yes, so he was asking, what do you mean that it's opt-out? So yes, because it's a trial in emergency situations, everyone is technically in, so because they give it during an emergency situation, so they don't need consent. And so if we want to not be included, we need to opt out. And so that involves a couple things like getting an opt-out medical bracelet. It involves writing it in your child's chart and making sure that they do not, if they don't respond well to Keppra, that they do not receive the Keppra.
So it's opting out of Keppra or opting out of ketamine, or both?
Both.
Okay.
Both. Yeah, both. But they know a lot more about it than I do, and they'll be here later during the cocktail hour to explain it, and then get contact info so they can send out some bracelets. But I wanted to make sure we were aware of it so that when they are here, folks can get those wristbands. Tomorrow, we'll be doing a pretty informal, I think, science recap at 9 a.m., talking about all the science that's talked about tonight and going over it using a little bit more family-friendly graphics and language, hopefully, because I'm not sure, if folks are going to tailor more to researchers or families, and we want to make sure that you guys understand everything that's presented at the gathering. And then here is a QR code with individual meetings with Dr. Hammer. So you can either scan this now, or I can send, or Casey can send, or Shelly can send the link out to you. But because Dr. Hammer's not here, he wanted to make sure that if people wanted to, they could meet with him. And so these are 30-minute meetings with him to go over the your child's genetic profile and any treatments or issues they might have been having, because Dr. Hammer is incredible. If you haven't met him, he's really, really wonderful. All right. Yeah. And then one more thing that Dr. Hammer wanted me to mention. Oh, yes.
Who's the sponsor of the cassette trial?
Of the KeySet trial, it is based at University of Virginia, but it's also all around the country. It's a multi-site. I think it's not just UEA, but the neurologist that I've been talking to is Dr. Jadeep Kapoor, who's at UVA, and then there's a couple other site investigators at different sites, but it is nationwide for the U.S. Yeah.
How does that delineate, like down through, how does that information like percolate, whatever, between the more rural or lesser known systems? Like, will all neurologists be informed?
Yeah, I'm not sure. That's a question I would ask them. Because Dr. Hammer's not here, he asked me if I could plug the SCN8A registry for anyone who hasn't filled it out. It's really, really important and helps us with SCN8A research moving forward. He also asked me to plug the SCN8A perspective study, which is seizure tracking, because we actually don't know the average seizure frequency of patients with SCN8A. And so both of these, if you can participate, the registry is a little bit lower effort. The perspective study is a little bit higher effort, but both are really essential for the future of SCN8A treatment, because this will help us get better treatments sooner than things like gene therapies. And while gene therapies are really, really exciting and life-changing, we want treatments now too. So